Chapter 12, Nervous System, Nervous Tissue, Part 1. So, the functions of the nervous system. Well, a little overview. There are three big category functions. The first is collecting information. The nervous system is constantly gathering information about both the external and internal environments. And it uses structures called receptors. And these receptors monitoring changes, say in blood pressure, in the receptors in our skin that detect if we are touching something or something is touching us, and collects and gathers that information, often known as stimuli. Information then gets sent to the central nervous system, where we go through the second function, which is processing and evaluating that information. Basically trying to decide if a response is needed for that information, for that stimuli. And then the third big category is if a response is needed, to initiate that response, to send out commands to structures called effectors. So initiating responses via nerves to the effectors. This can include causing muscles or muscle tissue to contract and or to cause glands to either start secretion or stop secretion. So the main effectors are muscle tissue and glands. Basically, the body's primary communication and control system. That is the function of the nervous system, to control and communicate with various parts of the body to maintain homeostasis, maintain that stable internal environment. When you look at the nervous system, you can break it up into two structural divisions. The first division is the central nervous system. The central nervous system consists of the brain and the spinal cord. Then the second division is the peripheral nervous system. The peripheral nervous system consists of the cranial nerves. These are nerves coming off of the brain, and spinal nerves, nerves coming off of the spinal cord, and also little bulbous structures called ganglia, which we'll talk about more later. You can also break the nervous system down into its functional divisions. You have the sensory nervous system. Sensory nervous system is where you find the receptors that are detecting stimuli in our environment, both external and internal. And then the sensory neurons carry that information to the central nervous system. So sensory nervous system is the nerves and neurons carrying the information to the central nervous system. Then within the central nervous system, we have the integrative functions, the processing and evaluating that information to decide if a response is needed. And then if a response is needed, it activates the motor nervous system. Motor nervous system are the neurons and nerves that go from the central nervous system out to the effectors, out to the skeletal muscle tissue, cardiac muscle tissue, smooth muscle tissue, and many various glands. Now this can be broken out even further. You can have the somatic sensory division of the sensory nervous system. Somatic sensory information is sensory information that we are usually consciously aware of. So sensations associated with our skin, with our muscles, with our special senses, vision, hearing, taste, smell. And then we have the visceral sensory system. The visceral sensations are the ones associated with our internal organs that we are not always consciously aware of. For instance, we don't know necessarily if the blood pressure has increased or decreased in a blood vessel. And then they send that information on to the central nervous system. And then you can break the motor nervous system down into two subdivisions. You can have the somatic motor division. Somatic motor neurons are the ones that control our skeletal muscles. So things that we voluntarily, consciously cause to happen, which is the contractions of skeletal muscles, skeletal muscle tissue. So that's the somatic nervous system. And then we also have the autonomic motor nervous system. This is the motor neurons that are going to control things that we don't consciously think about. The various organs and glands in our body that do the various jobs they do without us having to consciously think about it. All right, I'm going to move on to the main function of this, main topic of this chapter, which is the nervous tissue. So when you look at nervous tissue, there are two categories of cells. You have the neurons, which are extremely large cells. They're the basic functional and structural unit of the nervous system, and they are excitable cells, which means that they can transmit electrical signals along their membrane. And then the second category are glial cells. There are tons of glial cells in the nervous tissue. They are very small, so all these little dots represent nuclei for different neuroglial cells, and they are non-excitable cells. Their primary functions is to support and protect the neurons so the neurons can do their job. So, special characteristics of the neurons. Well, excitability, as we already mentioned, they will respond to a stimulus, which is usually the binding of molecules to specialized receptors, and that will cause them to generate a slight charge. So they respond to stimulations, basically molecules, neurotransmitters binding to receptors. That's going to potentially generate a charge. Then conductivity. Conductivity is their ability to have an electrical charge propagate along the membrane, moves along the membrane, going from, say, the cell body down the axon. This is called the action potential or nerve impulse, and it is basically very, very similar to the muscle action potential we talked about previously. Secretion. Neurons are able to release chemicals, specifically neurotransmitters, so that when that action potential reaches the end of the axon, they can release neurotransmitters. Those chemicals then bind to other cells, neurons, or effectors, and can be excitatory and excite the next neuron, or inhibitory and suppress it. Extreme longevity. Neurons exist for a very long time. Many of them, most of them, are formed before we are born and are still present when we become elderly. Also, neurons are amitotic, so almost all neurons do not divide. So most of the ones that we get from before we're born, we maintain and can't form any new ones. There are exceptions. The olfactory neurons for the sense of smell actually are able to divide. All right, components, pieces of the neurons. Well, we have the very large cell body. In the very large cell body, we can see the nucleus, very, very readily visible. The nucleus is the control center of the cell, so the control center of the neuron. Also found in the cell body are all the metabolic structures you need for a healthy cell. So the mitochondria, the Golgi apparatus, the smooth and rough endoplasmic reticulum, all those sort of structures are found in the cell body. And the cell body can also conduct electric signals toward the axon. So here is the axon. Now, the cell body is also considered an input structure, what this means is a synapse can form on the cell body, and at the sites of those synapses, the cell body's membrane has receptors that can bind to neurotransmitters, and that can generate the charge that then gets passed on to the axon. Finally, another thing found in the cell body is structures called chromatophilic substances. Chromatophilic substances are basically extremely active groups of ribosomes, both freely associated in the cytosol or bound to rough endoplasmic reticulum. These clumps of active ribosomes, it turns out, are grayish in color, and they are found primarily in the cell bodies, which means that if there is a part of the nervous tissue where there are a lot of cell bodies, that tissue is often grayish in color. All right, the second components are the dendrites. Dendrites are short processes coming off of the cell body. A cell body can have one dendrite, or more commonly has many, many dendrites coming off of it. The dendrites are primarily input structures, so they are the receptive ends to synapses. They have receptors that bind to neurotransmitters and can generate a charge that moves basically toward the cell body and then toward the axon. And of course, a neuron with lots and lots of dendrites has lots and lots of places where information can be received. And finally is the very long axon. The very long axon is usually a single process coming off of the cell body. The cytoplasm within the axon is called the axoplasm. The plasma membrane of the axon is called the axolemma. So those terms will show up during this lecture and future lectures, probably. It's a single, extremely long process. The beginning of the axon, as it forms off of the cell body, is called the axon hillock. So that is the very beginning of the axon. And axons can vary in length. They can be millimeters long, and they can go all the way up to 2 or 3 feet. You can have an axon stretching from the very end of your spinal cord going all the way down to the toes of your feet. So that is pretty long. The axon is the transmitting structure, so it makes contacts with neurons, muscle cells, or gland cells, and forms synapses, and then is able to release neurotransmitters to pass the signal, the information on to the next cell. And axons do not have chromatophilic substance. Actually, dendrites don't have it either. Now, when you get to the end of an axon, it branches off into lots of smaller processes. This branching off is called telodendria. So at the end of the axon is the telodendria that's spread out. At the end tips of each smaller process is a widened area called a synaptic knob. So all these little branches of the telodendria end in synaptic knobs. Synaptic knobs are filled with synaptic vesicles, which are filled with neurotransmitters. That should all sound a little familiar. Occasionally, you'll have an axon that splits off and has an axon collateral branch going somewhere else. This is relatively rare, but it can happen. All right, the cytoskeleton is extremely important for the neurons partly because they are so heavily branched out and because they have such long axons potentially. Cytoskeletons, as we know, made up of microfilaments, intermediate filaments, and microtubules. Specialized intermediate filaments, known as neurofilaments, stretch out from the cell body down through the axons. And these neurofilaments bundle up into thick protein-dense structures called neurofibrils. They're important because they provide tensile strength for the neuron. They make that extremely long axon strong enough that it is difficult to get damaged. It doesn't just break up on its own. All right, you can have fast axonal transport. Fast transport occurs at the incredible rate of 15.75 inches per day. It involves the microtubules of the cytoskeleton, and basically specialized motor proteins use ATP to move vesicles and organelles and things down to the ends of the axon. So that would be anterograde. And sometimes things using those specialized motor proteins move back up the axon, retrograde. So that used empty vesicles and potentially harmful chemicals and agents can get sent back up to the cell body to get processed. And slow axonal transport is a lot slower, 0.1 to 3 millimeters per day. Wow, pretty slow. And that is basically just diffusion and the slight flow of fluid, flow of the axoplasm. Doesn't require any energy. Things just diffuse and move gently down toward the ends of the axon. It could be enzymes, other cytoskeletal components, etc. And that is where we will stop for this part of the lecture.